I think everyone is out of the waiting room. Other people will find us along the way. I think we can start.
All right. So this is a very important webinar for us because we have been working on the plant course in the past few months. And today we are very honored to have Professor Jenn Brophy join us for this webinar. So the title could be a little bit different because I put this as a placeholder, but Jenn will be sharing with us about the synthetic gene circuit or the redesign of the plants in this webinar. So I want to do a little bit of introduction with Professor Jenn Brophy, who will be sharing us about the plant synthetic biology research today. So Professor Jenn Brophy actually graduated from MIT in her PhD. So, the lab that Jenn work at is like not too far from where I work right now. And then Jenn moved from the microbe engineering to the plant engineering in her postdoc and then joined Stanford as a professor and started diving more into the plant engineering and plant SynBio in her professor career. So I met Jenn at the in-person conference at the Asilomar conference. Kato was there as well, and then we chat about we're trying to do the plant course, and because Jenn is one of the leader in the field who do a lot of the plant SynBio, and Jenn is very excited that we are trying to do this and get this started. So after a while, we get in touch again, and Jenn is agreed, and we are very grateful that Jenn can join us for these kind of the wrap-up webinar for our Plant SynBio course. For those who are not part of the course, you're all welcome, and all the course materials will be made public in a month. So with that, I think this would be a very interesting talk from Jenn about, maybe a little bit about what is going on in the plant SynBio. Maybe we see some challenges that the field has been seeing and what we can do to overcome those challenges. And with that, I would like to welcome Jenn for the seminar, for the webinar today. I'll stop sharing and you can take it away, Jenn. Thanks a lot.
Okay. Thank you so much for that introduction and for the invitation to be here. As Ice mentioned, I'm really excited that you guys are getting into plants or considering doing synthetic biology in plants. So I'm hoping that there are several questions today. Please feel free to put in whatever you're curious about. I'm happy to have a chat about plants and synthetic biology in general. Okay, so I'm going to talk to you today about work in my lab where we're trying to engineer synthetic genetic circuits for cell type specific reprogramming of gene expression. Now, at a high level, my lab is interested in engineering the size and shape of plants, because in plants, like in all biological systems, size and shape are important determinants of function. And if we can figure out how to engineer the size and shape of plants, I think we can create some new and really useful plant varieties. On this slide, I'm showing two pretty famous examples in which people have changed the size and shape of plants through breeding. On the left is modern maize, which was bred from its wild ancestor, teosinte, to increase both the size and the number of seeds that the plant produces. And on the right is a dwarf wheat plant, which helped usher in the Green Revolution by allowing farmers to apply massive amounts of fertilizer to their fields and get an increase in grain yield instead of just more straw biomass. And what's exciting about these two examples is that the changes in shape are both the result of changes in DNA sequence. So as synthetic biologists, I think you can see this as an opportunity to apply some of these synthetic biology skills towards changing biological shape or plant shape. Now, we're really interested in modifying plant form because of how important it is for environmental stress tolerance. We're not really setting out to make a larger maize variety, although I do think that could be useful. We're interested instead in engineering the size of and shape of plants in order to improve their ability to withstand the environmental stresses that we believe are coming with climate change. So the shape and angle of a plant's leaves can affect photosynthetic efficiency and the ability of a plant to tolerate heat stress. And the shape of its root system can impact its ability to reach water and other essential nutrients in soil. So we're hoping that by manipulating these things, we can change their capacity for stress tolerance. Now, if that's the goal, then the challenge or question becomes, how do you actually engineer the size and shape of a plant? Well, one of the things that we know is important is really precise control over where and when within the plant you express developmental genes. To illustrate this point, you're seeing 3 different Arabidopsis plants, which we grew on these little agar plates in the lab so that you can see the shape of their root system. On the left is a wild type Arabidopsis, which has this long primary root that grows down in the direction of gravity, several root branches that also grow down in the direction of gravity. And if you were to zoom in on this root system, you'd see that it's covered in these tiny root hair structures, which are really important for water and nutrient acquisition. Now, a single base pair change in this plant's genome can alter several different aspects of its structure simultaneously. This plant in the middle doesn't make any lateral roots. Its primary root is shorter and has a hard time sensing gravity, which you can tell because it no longer grows straight down. It's also missing root hairs and has several structural changes in the shoot. And one of the reasons why this mutation affects so many different aspects of plant form is because the protein that's mutated is still expressed from its native promoter, and that promoter is active in many different cell types and tissues throughout the plant. Everywhere that this mutant protein is expressed, it's interfering with hormone signaling, and so you're messing with the kind of development of all of these different plant structures at the same time. Now, if we take that same mutant protein and we express it from a promoter that's only active in one cell type, in this case, it's in the stem cells that will later become lateral roots, we can make a targeted change in root structure. So in this plant all the way on the right, there are no lateral roots being made, but it's otherwise identical to the wild type plant. And so the strategy that we have in my lab is to really precisely control where we express genes in order to make targeted changes in the structure of a plant. Now, if you want to do this, you have several options, right? You can look through the literature for promoters that have expression patterns that match your desired expression pattern. If you work with model plants like Arabidopsis, there are several papers that have characterized tissue-specific promoters sort of painstakingly by one taking these promoters and putting them upstream of a fluorescent protein and then imaging the plants to see where is that promoter active. And you might be able to find a promoter in the literature that has the exact expression pattern that you want to execute your engineering goals. What you're seeing on this slide are just images of the root tips of some of these plants that people have made to characterize promoters. You'll see where the yellow fluorescent protein is expressed within the root tip. But I think hopefully what you can appreciate from this image is that these libraries of characterized promoters in plants are relatively small. They're only really available for some of our model plants, and even then, it's unlikely that you will have exactly the pattern that you want from a characterized tissue-specific promoter like this. So one other option may be to mine a promoter from an RNA-seq dataset. There are lots of RNA-seq datasets now that are either single-cell datasets or datasets derived from dissected tissues where you can get an idea of what genes in the plant genome are upregulated or downregulated in specific tissues. And you can, for example, choose one of those genes that has a pattern that you think is useful and use its promoter for your engineering purposes. Now, this is a good idea in theory. It's just in practice, it tends to be hard to actually implement. One of the reasons why it can be hard is shown on the slide. This is an example on the right where the authors of this paper took the 5' genomic sequence. So this was 2 kb upstream of the ATG of a gene called ARF7. And they fused what they thought was this promoter sequence to M. venus, expecting to see yellow fluorescent protein everywhere that ARF7 gene is typically expressed. But when they put this construct into Arabidopsis, they actually saw no fluorescent protein expression in the root tip, which was surprising because we know that ARF7, the gene, is expressed in the root tip of Arabidopsis. So when they tried troubleshooting this, they went back and they took not just the 5' genomic sequence, but actually the first intron and exons of this gene and fused that entire thing to M. venus. They then recovered the expression pattern in the root tip of Arabidopsis that they were expecting. Now, what that tells us is that there's information in this first intron and exon region that's important for driving ARF7 expression in the root tip tissues of Arabidopsis. And that makes it really difficult from an engineering perspective because you don't know exactly which portions of the genome are required for native genes to be expressed in the patterns that you observe, which means you don't know exactly which portions of the genome to use as promoters, as promoter tools for your engineering. All right, so if it's challenging, maybe if there are not to find promoters from the literature or to mine them from RNA-seq data sets, what can you do? One of the things, or what we try to do in my lab, and one of the options that you have, is maybe to use logic gates to generate new spatial patterns of gene expression. So I think you've already gotten a nice primer on logic gates from Lloyd earlier. And so I'm just going to tell you briefly about how we look at this problem from a tissue-specific patterns of gene expression point of view. All right, so on this slide, I'm showing you two different tissue-specific promoters. These are supposed to be cartoons of the Arabidopsis root tip. This one, this promoter is active in the root cap, which you can see from this sort of fake GFP pattern. And then this other promoter is active in a subset of root cap cells called the columella and in the central vasculature. So if you combine these two promoters' activities using a logical AND gate, you should get expression only where promoter A and promoter B are active or in those columella cells. And in theory, because there are actually 16 different two-input logic gates, 16 different ways in which you can combine two promoter patterns, you can make a large number of different tissue-specific promoter patterns from a small set of characterized promoters like this. All right, so this is work that was the generation of the first set of tools to create logic gates in plants that was done by me, was done in my postdoc. There are other people who have also built synthetic genetic circuit tools in plants. So because this work has been published, I'm not going to go into it in a lot of detail, but what I wanted you to know is that the logic gates that we built use synthetic transcriptional activators to turn on gene expression and synthetic transcriptional repressors to turn off gene expression. And by playing with where you turn on and turn off gene expression, you can create these logic gates in plants. If you have questions about how this works, I'd be happy to answer them. But what I'm just going to show you is two examples of how we use these logic gates to create new patterns of gene expression or to control gene expression levels in plants. So here's an example. This is one of my favorite logic gates. This is an A not imply B gate, so this is different than the AND gate that I showed you earlier. But the reason that I like this gate is because you can use it to refine a promoter's expression pattern. So here, input A is active in these root cap cells. This is the sombrero promoter in Arabidopsis for anyone who actually is working in plants already. And it has the same pattern as that cartoon I showed you earlier. And what you can do is you can use this promoter to drive expression of an activator. And then you can use a different promoter. This is the PIN4 promoter, which is active in those columella cells. And you can use it to drive expression of a repressor. And then you can have a synthetic promoter that's activated or repressed by these two synthetic transcription factors. And when you integrate their activity, you can actually create a promoter expression pattern that's refined relative to the sombrero promoter. So you can see here, you get expression in the lateral root cap cells, but not in the columella because it's repressed there. All right, so we think that that's exciting to be able to use tools to refine patterns of gene expression. You can also use these types of tools to control gene expression level in a tissue-specific manner. This is not trivial to do in plants. We know a lot less about how the sequence of a promoter impacts expression level. And so unlike in E. coli, where you can make certain mutations and have a pretty good idea of how that's going to affect expression level, we don't have that kind of control in plants. And so in order to try to tune how much gene expression you get in a specific set of cell types, we use these buffer gates. So buffer gates have a tissue-specific promoter driving expression of an activator, and then depending on how many binding sites you have in your synthetic promoter for this activator, you can get different levels of gene expression. Now, we used these buffer gates in order to try to engineer root branching in Arabidopsis. So I showed you before that we could totally delete root branching by expressing a lot of that mutant developmental regulator in stem cells for lateral roots. And we thought if we have this capacity to tissue specifically tune gene expression level, maybe we can make plants with variation in root branch density just by tuning down how much of that mutant developmental regulator we make in those stem cells. I should say that when we started this project, we didn't know what type of phenotype we would get. And it was also possible that doing this work would just identify a threshold above which we express so much of the mutant developmental regulator that we eliminate branching, and below which we get essentially wild type levels of branch development. We didn't know which of these things we got, but figured that a nice use of synthetic biology is also just to learn something about the biological system in which you're working. And so we designed this set of buffer gates in order to do this tissue-specific tuning of gene expression level. Just to be explicit about it, here is this tissue-specific promoter that's active in the stem cells, drives expression of an activator, and then we have a variety of different synthetic promoters, all of different strengths that are responsive to this activator, driving expression of our mutant developmental gene. When we put these into Arabidopsis, we were able to recover a gradient in root branch density. And what was exciting is that we did some analysis of these plants to show that indeed the only thing that was affected by the buffer gates, or at least the only thing that we measured that was affected by the buffer gates, was branch density. And so we've been able to really tune this aspect of plant structure. Now, what we're doing now is taking these plants with a variation in branch density and trying to understand how does root branch density affect things like survival in a challenging environment. So that once we decided, or once we demonstrated that we could control aspects of root architecture, a question became, what root architecture do you make, right? Great that you can modify this, but how do you actually use root architecture to improve plant stress tolerance? And it wasn't clear, for example, what root branch density we should recommend or go for when we try to improve plants' tolerance to environmental stress. But now we had these plants with variation in this feature that we could use to try to answer this question. And I think that this type of analysis, let me skip ahead actually one slide just to show like this is how we think about this problem. We're trying to use synthetic biology to build plants with new structures that then we can study in order to understand the contribution of that structure towards plant survival in a challenging environment. And that's not easy to do. And if you look at the other methods that people use in order to study changes in structure, you'll find that often people will look at a diverse population in order to get variation in a feature of interest. So these are hummingbird wings. And to understand how the shape of the hummingbird wings affects flight, people will look at different hummingbird species because then there's variation in wing shape. And they'll draw conclusions about how that wing shape is affecting flight. However, there are several different aspects of the bird that also change as you move from species to species. And so it becomes a little bit more challenging to attribute a change in flight, for example, to exactly that change in wing shape. Analogously, right, the same type of thing is done in plants, where as you put together a population of plants with variation in some feature, that it becomes challenging to attribute any difference in survival to that feature. To kind of circumvent this, people have started simulating variation in structure with models. And so you can model different root architectures, for example, and try to predict which ones will be better at absorbing water or nutrients in different types of simulated soil environments. But obviously, it would be nice to validate some of these model predictions experimentally. And we think that that's where synthetic biology can really play a big role in understanding form and function relationships. Okay. I just want to point out or take a second to say that studying relationships between form and function in plants is not that easy, just because plants are really sensitive to the environments in which they grow, and they have this ability to change their size and shape, their form in response to the environment. This is a picture that I stole from Twitter, which shows a tomato plant growing outside in full sun. And you can see it has this sort of short, bushy feature, structural, it has this sort of short structure. And then if you take this same type of tomato and you put it into a low light quality environment, like this cardboard box that has this kind of maze-like thing inside of it, you'll see that the plant starts to elongate its stem. And it's searching for higher quality light. And what that means is that it's really difficult to ask questions about how stem height affects the plant's ability to survive in a low light environment, because as soon as you put it into the low light environment, it starts elongating that stem. And we think what would be really useful is if it always had the same stem height, kind of regardless of what environment it was grown in, so that you can isolate the impact of that stem height to survival in a specific environment. How we are trying to use synthetic biology to do that type of isolation, to try to sever the tie between form and the environment, because we think if you can cut off all of these different inputs that try to influence how the plant grows, then you grow the same shape all the time and then see what the impact of something like a specific root structure is on the plant's ability to survive in a given environment. Now, we have one of the reasons I bring this up is because the way that we're modifying form is through these mutant auxin response regulators. That mutant protein that I mentioned is a hormone sensor, and in response to high concentrations of a hormone called auxin, it will initiate the expression of different developmental genes. And at a very high level, all of these environmental signals influence auxin biosynthesis and degradation, response, and the way it's moved throughout the plant. So in theory, if you remove the impact of auxin on developmental gene expression, you may become less sensitive to the environment. So we've done some preliminary work showing that the plants that we've made with variation in root branch density have consistently this gradient in root branch density across different environments. And that even though branch density changes across these plants, other features of the plants are relatively consistent across environments. That's allowing us to do experiments like this, where we try to attribute or correlate the health of the plant with how many root branches it has. So this is a sort of control experiment. These are really preliminary data. So it remains to be seen as we increase the replicates here, whether these relationships are real. But what we've seen in some preliminary experiments is that as we increase root branch density by having plants with different synthetic genetic circuits in them that allow for more or less really branch development, that the more branches leads to larger rosettes, larger leaf above ground biomass, essentially. And that makes some sense, because in order to be able to grow a larger rosette, you need more nutrients, therefore having more branches can be helpful. But in salt conditions, which we tested because as lands start to dry out, the solute concentration goes up and they become saltier. We find that plants with intermediate levels of root branching tend to have the largest shoot biomass. And we're still trying to understand this, but it could be that, as you have more root branches, you're taking in more nutrients, but you're also taking in more sodium ions, which can be problematic for the growth of the plant. And we're trying to quantify the relationships between branch density and the survival in different environments to try to have this quantitative understanding of how these feature effects develop. Okay, so if you're like a huge plant nerd, maybe that was exciting to you. If you're maybe not as much of a plant nerd, we're going to get back to some more synthetic biology in a second. First, I have a little bit of a warning, right? So I'm not sure how many of you have done experiments with engineered organisms, but you'll find that sometimes they do things that are surprising and maybe unintended. And in this case, what we've found by growing our engineered plants for long periods of time or under stress conditions is that occasionally they have this surprising behavior. So these are plants that we've engineered to have no root branches at all. You can see on the control, it just has this one long primary root. This other root that you see coming out, this is called an adventitious root. It's actually a shoot-borne root that's not something that we have tried to control the growth of, and so occasionally you'll see them grow. When we grow these plants with no lateral roots, occasionally we find that the primary root tip detaches from the media and kind of grows out into the air. And when it does that, the root tip will die. And when it dies, we will see this explosion of lateral roots near the site where the primary root had died, which was really surprising to us because we thought, oh, these plants don't have the capacity to make lateral roots. So to see this big explosion was really quite strange. The other thing that we observed is that when the plants are grown under stress conditions like salt, sometimes they will also start to make lateral roots. Again, not a phenotype that we expected. Now, when we started digging through the literature for potential causes for this type of response to either primary root death or stress, we found that there are actually two different lateral root stem cell specification pathways. There is the canonical one where you get high levels of this gene called GATA23 being expressed, which marks cells that are then competent to become lateral roots. And we used the promoter of GATA23 to express that developmental regulator that eliminates root branching. So every stem cell specified by this canonical pathway, we had suppressed the development of those lateral roots, right? But there is this alternative method of getting lateral root stem cells that is triggered by a gene called WOX11 in response to wounding or stress that can also lead to lateral roots. And we were not suppressing this pathway. We were not engineering it, in part because when we did this work, we were not aware of it. But the synthetic the circuits that we introduced kind of revealed the importance, at least to us, of WOX11 and its role in root development. Okay, so what we would like to do now is try to delete this pathway and make it difficult to produce lateral roots through WOX11 so that we could really control how many lateral roots these plants were producing and not worry about them creating a bunch of lateral roots under stress conditions. The problem is that this WOX11 gene, you cannot just use it to express something like that mutant nsAA that represses auxin signaling, because this promoter of WOX11 is on in those stem cells, but also on in the root tip of Arabidopsis. And so if we suppress auxin signaling in the root tip, we know that several bad things happen to the plant. And what we would like to do is just suppress auxin signaling in the WOX11 derived stem cells. And we think that the way to access these stem cells is to apply an AND gate where we have WOX11 signal and a signal for the type, the cell file in which these stem cells typically appear, called the xylem pol paracycle. And this requires using an AND gate to combine these two promoters' activity. The AND gates that we had previously made are quite complicated, and so there's a graduate student, Isabel, in the lab who has been developing new split transcription factor-based AND gates. All right, so splitting a transcription factor into two pieces could give you an AND gate, because just then whenever you have both fragments expressed, you get reconstitution of the functional transcription factor, which can then drive expression of your output gene. Now, TET-R, which is a type of bacterial DNA binding protein, had been split in previous work. In this case, this is a split from a paper in ACS Synthetic Biology, and shown that the two halves could be brought back together to create a functional transcription factor in E. coli. And so we tried using a split TET-R in plants. We have a promoter that responds to TET-R by being shut off. So typically this promoter is on, and then whenever TET-R is expressed, it shuts back off. You can see the full-length TET-R shuts it off quite nicely. We then tried splitting TET-R into two pieces exactly where this paper had split it. And we have this nice kind of not and design where you only get repression of that promoter when both pieces of TET-R are expressed. So this tells us that these two halves can find each other in plant cells and then repress gene expression. There was some pushback here at Stanford when we first had this idea. There was a lot of skepticism that two pieces would be able to find each other in the comparatively large space of a plant cell. However, there had been a paper published in plants showing that you could build ANGate with split transcription factors from Mauricio Antun's lab. So we were pretty confident that this would work. Now, TET-R is a great DNA binding protein, but we wanted to see if we could split some of the synthetic transcription factors that we had been using already to construct logic gates in Arabidopsis. So most of our synthetic transcription factors were based on TET-R homologs. We tried splitting these and, we tried aligning these to TET-R to see if we could just split them in exactly the same location to create lots of split transcription factors that we could use for synthetic genetic circuit construction. But the alignment of TET-R with these homologs was poor near the known TET-R split site. So Isabel started trying to split her synthetic transcription factor in a number of different locations and found that some locations, her transcription factor could be split in half and then reconstituted quite easily. What you're seeing on this graph here is two controls. This is a promoter that's activated by her synthetic transcription factor. In the absence of the synthetic transcription factor, it's got pretty low GFP. With the full-length transcription factor, it has this increase in GFP. And then each of these are showing you one half of the split transcription factor or both halves together. This first one here, the split site is non-functional. So even when you express both halves, they never come together to produce a functional protein. But for some of these other locations, she was able to show that the transcription factor is functional when they come back together. So we thought this was exciting. And what I'm not showing you is exactly where the split sites are, but some of them are quite early in the protein and some of them are quite late. And Isabel has been able to show that you can actually use this information to split the protein into 3 pieces and get 3 input AND gates that function well in plants. And she's currently working on extending this to see how many times you could split this transcription factor into pieces in order to create these really high order logic gates. I had like a whole bunch of other stuff to talk about, but I think what I'm going to do, given how much time we have, is skip to a story about bacteria, or should I stop here, Ice? Would you prefer?
I think you can keep going. I think it's very interesting to me right now. I think that would be for
Okay, just didn't want to take
audience as well. Yeah.
If it starts to go too long, just cut me off. Okay.
Yeah.
Okay. All right. So we're gonna skip to a story about bacteria. So there's been work showing that microbial treatments or the interaction with plants and their microbes can be really important for plant health. There have been some really interesting studies showing that soils from arid environments can be applied to plants that are not from that arid environment to protect them essentially from drought stress. And it's believed that it's the microbes in that soil that are important for that drought protective effect. So there's a great postdoc in the lab, Kate, who has been trying to see if she can design collections of bacteria that help protect plants from drought stress. And I'm not going to tell you about how she's been designing those communities. This is just an example showing like wheat plants that she's grown that were well watered versus droughted, or droughted with a synthetic community of bacteria that are supposed to be protective from drought. It does look like some of these communities that she's building do have this drought protective effect. But we have a lot of work to do still to figure out, how the community is working and what design rules exist for trying to put together a community that has a function like this. But one of the questions it's raised for us was how, if we can create these communities of bacteria that help plants survive periods of drought, how do we limit that benefit to specific plants? This is a picture of soybean plants growing in rows in the field, and you can see the massive number of weeds that are also growing in these fields, and these weeds use resources that you're trying to apply to the fields to help your soy plants grow, right? They're using water and micronutrients in the soil, and it would be nice to try to support just your crops of interest so that those resources can be stretched farther and have more of the intended impact. All right, so what we have been considering with a collaborator at the University of Delaware, who I'll show you in a second, is whether or not we can send some sort of signal to the bacteria that tells them this is a plant that you should help grow. And that signal would be, contained essentially to the plants that we want to help grow, and that the bacteria would then have very little plant growth promoting effects on other plants that they encounter, like weeds. And the signal that we are trying to use for this work is a non-standard amino acid. So there are 20 amino acids, right, that you can use to build proteins, but then there are, I think, close to 500 examples of non-standard amino acids. So these are sort of modified versions of the standard amino acids that are not typically used by organisms in order to create proteins. And we thought if we can secrete one of these non-standard amino acids to a bacteria that's engineered to use the non-standard amino acid for production of a specific plant growth promoting phenotype, then maybe we can link plant growth promotion just to specific plants. Now, the process of engineering bacteria to use non-standard amino acids to create specific proteins is called genetic code expansion. And this works by typically inserting a stop codon into a gene that you would like to express only when there's non-standard amino acid present. So this is a nanoluciferase, which has a stop codon in it. You make the mRNA of the nanoluciferase all the time, but you cannot translate it typically because there's the stop codon that stops protein synthesis after just a couple of amino acids. In this bacteria, what you would also put in is a tRNA that's been designed to bind to this stop codon, so that now this stop codon, instead of recruiting release factor that terminates peptide synthesis, you recruit this tRNA, and that tRNA gets charged with a non-standard amino acid by a synthetase that you also introduce to the bacteria, so that it puts the non-standard amino acid into the protein as it's being made and allows you to synthesize the entire protein. But importantly, this only occurs when the non-standard amino acid is available, right? Without it, you should not be making this nanoluciferase. Okay, so here are our fabulous collaborators. We have been working with Aditya Kunjapor's lab at the University of Delaware and his students, Michaela, Amanda, and Avanik, because they have been able to engineer Bacillus subtilis, a type of soil bacterium, to require non-standard amino acids for synthesis of chosen proteins. So in this case, this is GFP that has a stop codon in it, and you can see strong expression of GFP only when the non-standard amino acid is present in the growth media. And for this work, they've been using a non-standard amino acid called O-methyltyrosine. So this is just a methylated version of tyrosine, which is a standard amino acid. You can actually synthesize O-methyltyrosine through a single enzymatic step using a methyltransferase called MFNG from a Streptomyces strain. They had figured this out previously, and so they asked us, could you try putting this gene into plants to see if we can engineer plants to create O-methyltyrosine? And so a graduate student in the lab, Vivian Zhang, has been doing this work of figuring out whether we could engineer plants to create O-methyltyrosine. The first thing she did was a transient expression of the enzyme in Nicotiana leaves. She delivers the DNA encoding the enzyme to plant leaves using agrobacterium, and then took samples of the leaves and ran LCMS where she was able to show that the infiltrated leaves, the leaves where we've delivered this MFNG gene, are able to make O-methyltyrosine. Here's the standard, and that the plant does not naturally produce this non-standard amino acid. So this was exciting to us because it showed that there was capacity for making O-methyltyrosine in plants. We have started making Arabidopsis lines that express this enzyme, and this is an example of an Arabidopsis line that expresses the enzyme from a ubiquitously active promoter, so in all cell types in the plant. And we've been doing, or Vivian has been doing assays to figure out how much O-methyltyrosine these plants actually exude. So she's been growing plants on little filter rafts in hydroponic growth media. This, I believe, is a 96-well plate, just to give you a sense of scale of how small these are. And what she'll do is she'll grow the plant in this hydroponic growth setup and then collect the media and run it on an LC-MS in order to figure out how much O-methyltyrosine is being secreted by these plants into the environment. And it is quite a small amount. So like nanomolar concentrations of O-methyltyrosine end up in the media here. But that was enough for us to try to test whether or not this trans-kingdom signal could be detected by the bacteria. So she ran experiments like this, where we have the engineered plants growing on agar plates. She would drop her on a little bit of the bacteria and then wait a couple of days for the bacteria to sense the non-standard amino acid signal from the plant, and then she would phenotype. In this case, the bacteria are expressing a luciferase in response to the non-standard amino acid, and so she's dropping on the luciferase substrate before taking luminescence measurements. So here's an example of what these experiments look like. This is a constitutively active Bacillus subtilis, so it creates the luciferase regardless. It has no stop codon in the luciferase gene. And you can see a luminescent signal on either the wild type plants or the plants that are producing the non-standard amino acid. But for Bacillus subtilis that do have that stop codon that require O-methyltyrosine in order to make the luciferase, we saw a strong signal only when the bacteria were grown with the plants that secrete the non-standard amino acid. So this was probably the first assay that Vivian ran with this plant to test this plant microbe interaction, and I was pretty surprised that it worked. But one of the things that's important here is to look at how unhealthy these plants are. They're very small. These are the same age as the wild type plant, and their growth is very stunted. So the plants clearly do not, although they can produce the O-methyltyrosine, they do not like it. So she's done a lot of work to try to figure out maybe how the O-methyltyrosine is impacting plant growth. It looks like there's a defect in germination rate or time, how long it takes to germinate the seeds, and how rapidly the roots grow. And as she was trying to figure out how O-methyltyrosine was impacting plant growth, she was also trying to find a solution. Because obviously, if your plant is unhealthy, just making the signal that signals to the bacteria, hey, I'm the plant you help, then that's not very useful. And the plant itself needs to be healthy in the first place. So one of the things she tried was to restrict where within the plant the O-methyltyrosine was being made. Instead of expressing the enzyme ubiquitously all throughout the plant, she started to use this root cap tissue-specific promoter to express the enzyme that produces O-methyltyrosine. And we chose root caps because that's a site of exudation of a variety of things, including carbon and amino acids, into the soil. And so she thought maybe if we just synthesize the non-standard amino acid here, we won't be using so much of the tyrosine that the plant otherwise needs to grow. And if the non-standard amino acid itself is toxic, we won't see that effect either. But we should hopefully still be able to be putting enough of it out into the environment to help the plants grow, help the bacteria. All right, so she made those lines where it was expressed just in the root caps and then tried these co-cultivation assays again. The assay setup has changed a little bit. In this case, we have started mixing the bacteria into the agar plate and then plating the plants on top of this agar that has bacteria mixed in, because the dropper assay was not giving us a good picture of kind of everywhere that the bacteria were able to sense the non-standard amino acid. So we would cold cultivate these things, and then she would pour a thin film of luciferase over the entire plate and image to see where the bacteria were detecting non-standard amino acid. So here's an example where we have a constitutive bacteria control. So these don't require the non-standard amino acid. You can see a strong luciferase signal all over the entire plate. Here's a negative control where we have the O-methyltyrosine dependent bacteria growing with a wild type plant, and you can see no luciferase signal. But then here's the ubiquitin lines. So you can see that the bacteria are able to sense this non-standard amino acid being made by the plant. And then here's the root tissue specific line. You can see the overall signal is much lower, so the plants are making less of the luciferase in this case, or sorry, less of the O-methyltyrosine in this case, but still enough for the bacteria to at least detect and produce this luciferase signal. The rest of this has been centered around trying to change the output gene from a luciferase into a gene that's essential for the bacteria to do a type of biocontainment, so that the bacteria cannot grow, not just can't produce a luciferase, but can't grow unless the O-methyltyrosine is present. And to sort of spoil my own talk, this doesn't really work. The bacteria, though they look like they require the non-standard amino acid when we do assays with the bacteria in growth media, this is without the O-methyltyrosine in the growth media, and then with it or the wild type bacteria, both of those grow quite well. The dependence of the bacteria on O-methyltyrosine when we start to co-cultivate them with plants seems to disappear. So I just have this, like, life finds a way, biocontainment is very hard type of takeaway from this. There is a lot of engineering that our collaborators are interested in doing in trying to improve the biocontained strain so that it really is dependent on the O-methyltyrosine for growth in these kind of co-cultivation conditions, but we're not quite there yet. Okay, so in general, I would like to push for synthetic biology in plants to start to try to realize this next generation of crops where we move from traditional overexpression lines where a single gene is expressed everywhere within the plant throughout its entire life cycle, to plants where we express genes just in specific tissues or in response to specific cues, because I think that we can access more useful and interesting phenotypes by controlling where and when they're expressed. And so I'm hoping that we see a kind of blooming of synthetic biology in plants, that you all get into this space, because I think there's a lot to do here. With that, I just want to thank the lab. I tried to highlight the people whose work we talked about here, but everyone in the lab is absolutely fabulous, including Renee, who has been a part of this program this summer. And if there is time, I'm happy to take questions. Sorry, that was long.
Thanks a lot, Jen. That's a great, overview, and I really appreciate you going through the non-canonical amino acid discussion as well, because it might be new, and not all the time people will take time and explain what exactly it is and how exactly do we get integrated into the system. So that's a really nice explanation right there. We do have time. If you can stick around for maybe 15 or 20 minutes for the question, it would be perfect. We have some question in the chat. And I'll go through some questions that were available right there. But in the meantime, if any of you guys in the audience are interested, or have questions, please feel free to raise your hand and you can sort of speak up yourself as well. So I would just try to go from the bottom to the top so that we kind of get into the discussion that we just mentioned and get back to the first part later. So we have a question from Nana about the tRNA. So for the tRNA that binds your stop codon, like how is it specific for the non-standard amino acid synthesis? Like, will the normal amino acid transcription also, being affected if you sort of do that? I think this is more of the question on the microbe side. So if you have some information from Aditya's work, that would be great to expand a little bit more on that as well, Jenn.
Yeah. So it's a good question. The specificity of incorporation of the non-standard amino acid is dependent on the synthetase. So the synthetase is the enzyme that tacks the non-standard amino acid onto the tRNA, and then that tRNA will bind to the stop codon so that the amino acid can get incorporated into the growing peptide chain. Now, what we found is actually that the synthetase, though it can attach O-methyltyrosine, probably also with a low frequency attaches standard amino acids. It's one of the reasons why the control is potentially not as tight as we might like it to be. I think the other portion of that question was about how it's affecting other genes, because there are many stop codons in the Bacillus subtilis genome. We are specifically targeting the TAG stop codon, which is the least common, this amber stop codon. But it will, like, having this tRNA in the bacteria will mean that many of the proteins that the bacteria is trying to make will not stop at the place where they usually stop. They will have this non-standard amino acid incorporated. And so this system does mess with the proteome of Bacillus subtilis. There have been many efforts in E. coli, at least, to do engineering of the entire genome to remove all instances of TAG stop codons so that you can have a system like what I described today without interfering with the expression of native genes as much. But that hasn't been done for Bacillus subtilis yet. Hopefully that's clear.
Yeah, which I bet it was still ongoing, and at some point we will have that around. I think there are several groups trying to make that happen for the community as well.
Yeah, genetic code expansion is like a big thing.
Maybe I'll touch on the question from Johanna, which is like a little bit relevant to the part of the syncom that you were showing in the earlier of the microbial talk. So the question reading from Johanna is like, how can the beneficial plant microbiome protect plant under those conditions of climate variability? So maybe like if we go back to the drought condition that you show, can you elaborate further, like what exactly they're doing so that they kept the plant from, severe growth effect from those things? And maybe another thing that might be a little bit relevant, I'm not sure, is, can we, repurpose these for some kind of like metabolite production when we try to use plant for the signaling between microbe and plant? So these are two separate questions. Like can we do something to make them make the metabolite sustainably? So maybe we can go back to the first question first about what is going on there.
Sure. Yeah. So I think there's two questions here. So I think there's two questions under like just how do we have microbiomes that protect plants from severe environments, and then how do we make sure that that capacity to protect plants is preserved even though the environments are fluctuating. For the wheat drought project, one of the ways that we started to try to address this was to think about what happens to a wheat plant during periods of water deficit stress. When there's not enough water, what happens to the plant? Obviously, it needs water for turgor pressure, but it also uses a process called transpiration to draw nutrients up from the soil into the above ground portions of the plant. And as you become droughted, you start to close the stomata in the leaf. And as you close the stomata in the leaf, you reduce transpiration. And so you can start to reduce the amount of nutrients that the plant is able to acquire. So one of the things that we thought was, well, maybe it's becoming sort of nutrient limited. Maybe if we can create biofilms around the root system, that we could help protect the plant roots from kind of leaching water back out into the soil environment, because that process can run in reverse. And we didn't really know, but we have this very large library of microbial isolates, which we started running through these high throughput screens to see what types of kind of beneficial phenotypes they might have. And then the way we've been designing synthetic communities is to try to test the contribution of each of those potential features towards like drought protectiveness. So we were trying to design the communities to say like, okay, if we drop out the ability to produce biofilms, does that reduce the effectiveness of the community? And can we start to say like, these are the features that are important for that drought protective phenotype? I'd say like we don't really know. There's a lot of issues with just like stability of the community, right? There's been a lot of work to try to figure out how different collections of microbes can stably grow together without one sort of taking over the population. And that's hard to figure out for one condition. So, as you think about conditions varying, it becomes even more challenging to do. So I don't, I don't have the best answer to this question. One way, I'll tell you that like one way we've been interested in tackling this is by looking at how the root exudate profile changes in the plant over time and under different conditions. So we know that as the plant, like when the plants are really young, they tend to secrete a lot of sugar into the environment. Like there's this theory that it acts as a beacon and recruits a bunch of microbes, and then that as the plant ages, it starts to reduce the amount of kind of more broadly digestible carbohydrates into like organic acids and things that are maybe more selective for specific members, and that under stress conditions, you can see the profile of the exudate change. And so there's some theory that like, maybe you could design a community so that the composition of microbes is able to capitalize on the shifts in what is available to them in order to make sure that microbes that you think are protective are going to be able to, if not grow, at least survive as the conditions become harsher. But it's a really complicated space, and it has honestly taken Kate into a much more microbial ecology kind of world than I think either of us were expecting, where there's a lot of unknowns.
Yeah, that that makes a lot of sense. I think so for those who are taking our class as well in the audience, we have like one lecture that we're focusing on the plant microbiome or some kind of like manipulation. Big, who is also in this call, was the one who were part of the instruction on that class. So if you want to listen more about this kind of like drop in, drop out in details, you can take a look in that as well. I think this is a complicated setup, and Jenn has been walking us through what is going on in this specific drought system. But there are a lot more things that we have to consider when you design the actual experiment. Yeah, so thanks a lot for that detailed answer, Jenn. Maybe I want to touch on the second question a little bit. So this is more of the general question. So maybe I'll frame it in like, what would be the regular or, common tricks if we were to make plants or convert plants into the cell factory to produce metabolites in plants and like make it more sustainable in some ways? Because usually we use microbes because it's fast growing. That there are some other interests to use plants because it might be even easier to grow in the field for some reasons.
Yeah, that is such an interesting question. So I'll preface this by saying like I'm not a metabolic engineer, and I have, as a kind of general rule, tried to stay away from metabolic engineering. In part because it's hard for me to wrap my head around how you can dramatically increase production of a given compound or protein or whatever it is you want to make without some sort of drawback, right? Without compromising something else. And, we have a project with the CABI, one of these DOE centers right now, to look at an oil cane variety that they've developed, where they have transformed sugar cane to try to increase the amount of oil that the sugar cane is producing so it can be harvested for biofuels. And they've published papers on this. You can read them. They're using this push-pull protect type of approach where you increase, the amount of precursor to the oil that's being made by transforming in an enzyme that helps, push flux through that pathway that gives you the precursor. Then the pull is the actual transformation of the precursor into the desired oil component, and protect is the actual like deletion or downregulation of enzymes that would redegrade the oil. And this works. You can actually pretty dramatically increase the amount of oil that the plant produces. However, there's yield drag. The plant is a little bit less healthy, sort of like we showed you with O-methyltyrosine. There is a side effect to producing a bunch of this oil. Now, O-methyltyrosine we think is toxic just in and of itself. And so in addition to maybe siphoning off tyrosine resources, the production of the non-standard amino acid itself, like that molecule, is problematic. And I think there might be ways of engineering the plant to not feel the toxic effects of the non-standard amino acid so much. And with the oil cane project, we are looking at like, what is the mechanism of yield drag? Is it that the push, pull, protect is actually like depositing a bunch of oil in places within the plant that's problematic for the proper functioning of the plant? Or is it that it's siphoning off resources? And I think, if it's putting it in certain places within the plant, that might be possible to overcome. But if it's the siphoning of resources, then I'm not sure, right? But I do think that sustainable production of molecules in plants should mean that the plants still grow well. You have the sort of same use of resources to grow the plant that's wild type versus the plant that's producing your molecule of interest. And that there are a lot of engineering challenges in getting like a high producer that's really healthy. But that is my really naive understanding of this, not having thought as much about metabolic engineering as I know a lot of people have. So.
Yeah, there are some other notes that I heard from other speakers about like the plants has different parts into the trees, and it might be non-trivial to engineer those accumulations of metabolites in the certain parts. Like let's say if you want it to be more in the fruit rather than in the roots, that could be some other sophisticated engineering as well. So I think that's what I heard, that it was hard. I don't think anyone know how to efficiently do that yet, as far as I know, but maybe soon if you know more about plants in bio, right?
Yeah, we've been interested in there's a question of whether there's a growth defense trade-off in plants because they can produce a bunch of secondary metabolites that are bitter or toxic to herbivores, or can serve as sort of a deterrent for predation. And the signaling molecules that seem to stimulate the overproduction of these secondary metabolites also seem to stunt growth. And it's sort of unclear, at least by our reading of the literature, whether those two things are separable, right? Like whether it is possible to synthesize a bunch of this stuff while continuing to grow well, or if there's a reason to have a growth defense trade-off like that. So, I mean, synthetic biology, I think, could be used to answer some of those questions.
Thanks a lot, Jen. Just in case any others have questions, please feel free to raise your hands. We have like maybe one or two in the chat right now. I have some that I take notes, but I'll prioritize the ones from the audience first. So there's a short question from Innocent asking, can we engineer, or we can have plants that can grow in some kind of like acidic or salty or alkaline condition? How far are we on those kind of engineering trajectory?
Yeah, so it's a great question. I think it depends on what the goal might be. So there are already plants that can grow in acidic or salty or alkaline soils. The question is like, do we want to figure out how to grow some of the crops that we really rely on for food production, for example, in conditions that are not ideal for them. So like wheat, for example, doesn't do that well in saltier soils. So is there a way to learn from sort of the plants that do grow well in saltier soils and use some engineering to kind of move that capacity over into our like chosen crops? And, I'd say I think we're making progress in that there is a combination of like we need to understand a little bit more about how plants are able to tolerate those conditions and which mechanisms by which they tolerate them might be portable from one plant to another. But I don't know of examples where that's already possible. But I think that's one of the reasons why we need more work in plants, right? We need more of this ability to, we need more studies of plants to understand like how the plants that do grow in really salty conditions are able to do that. And then more engineering tools developed so that as soon as you know how that works, you're like, great, now I have all the tools already in place to translate that knowledge. And I think, all of that would be great to keep pushing forward.
Excellent, Jen. I think we have a question from Comfort who raised his hand. So maybe you try to speak up, Comfort?
Thank you, Ice. And thank you, Professor Jenn, for a very inspiring presentation. A very nice one. And my question is a little off. It's related to career. If I needed to join your research group, are there like opportunities one can apply to or something of the sort? Thank you.
Yeah, great. So thank you and hello. So I don't know what career stage you're at. So I'll maybe rattle off different types of opportunities. We have for graduate students at Stanford, we do general admit program. So you can apply to any of the biosciences programs at Stanford and then rotate through any of the biosciences labs essentially on campus. So even though I'm in the bioengineering department, we have students from biology, and we could in theory take students from any of the biosciences, biophysics, genetics, and stuff like that. So I would encourage people anyone who's interested in doing PhDs to reach out that way. We don't have the capacity to do direct admit, so it's not possible for me to take a PhD student into my lab who is not going through one of those programs. That being said, we have hosted summer students from other universities and even other countries in the lab. It's really helpful for those to set them up pretty far in advance. So I'd say if you're interested in like visiting for a summer to do research, reaching out in the January, February timeline for summer is helpful. It just takes so long to get through all the bureaucracy at Stanford. We are potentially able to take on maybe one more postdoc, but we have a pretty large lab already at this point. And so far, no one has graduated yet, but we're hoping that changes soon and we start to, get onto a cycle where we're able to take more people into the group. All of that being said, right, if that's the career stage, then the process for that is just to reach out directly to me, and we can have a conversation about whether there's interest in the types of projects where we do have some funding support available. That being said, I am not the only person that does a lot of plant synthetic biology, and I am happy to also just provide recommendations for other groups that are in this space who may have either a better fit or more opportunities at the moment. I don't think it's a secret that things in the US at the moment are pretty unstable as far as research funding goes. So it's been a slightly harder year to make predictions about what kind of capacity for new opportunities we have. But I'd be happy to help people who are interested in plant synthetic biology brainstorm how they could get there, whether it's with me or somewhere else.
Okay.
Thanks a
Thank you.
lot, Jen.
Thank you.
And thanks a lot, Comfort, for the question. So maybe I would add more a little bit onto Jen's answer, is that like we have one webinar that was dedicated to like how to apply to a PhD. We use the US as a case study, but that could be applicable to Europe or in other spaces as well. And to echo what Jen said, that there are growing community in the plant synthetic biology and in the synthetic biology system as well. So for our past speakers, we have James Lloyd, who just established his lab in University of Western Australia. So he actually has like, a post about he's looking for postdocs and also students as well. So, there will be time and cycle of these applications. So if you're interested, maybe we would suggest to go to the webinar that we have, take a look at our website. We have one from Malik and Miles who were a student and walk through that themselves. So that would be a very interesting process to understand how the application goes. With that, I wanna like get back to one of the question that was still in the chat, and maybe we can do maybe like 2 or 3 questions before we wrap up. Hopefully, Jen can still stay with us until then. So there was one question about the about the redundant pathways of the plant that was asked earlier about the plant SynBio to begin with. So if I read the question, it was like, in what ways can manipulating plant genes can help reveal the function of the redundant pathways or something like that? So this is like probably rooted from some of the conversation that we had during the class, and we know that there is a lot of genes in plant. There are even some genome amplification or other things that happen in the plants. So maybe Jen, can you share with us, can we use anything on this plant engineering or plant SynBio to help explore these kind of redundant pathways, or is it still difficult?
Yeah, I mean, so I'm going to give you a biased answer, which is like our experience with finding redundant pathways. The one example that I presented today with lateral root stem cells, I think can be used to answer this question. We set out to try to control how many lateral roots those plants were making, and we used a promoter that should be active in these lateral root stem cells to express a gene that would essentially inhibit the kind of further development of stem cells into a lateral root. And by doing that, what we found was that there's actually this other pathway that's also active in, producing lateral root stem cells that have the capacity to then make lateral roots. And, we weren't the first to discover that. That was already in the literature. But as we try to delete that other pathway, one of the conversations that we've had a lot is what happens if we suppress lateral root development in that pathway and the plant still makes lateral roots. And I think in that case, we would end up in a space that's pretty uncharted. And in some ways, it would be frustrating because we wouldn't know, we wouldn't have achieved the engineering that we wanted to, but in other ways it would be exciting because then that would be potentially plants we could use to identify a previously unknown redundant pathway, right, for producing lateral root stem cells in this case. So I do think that like the process of engineering in plants starts from, knowing a lot about plant development. It's really helpful, or plants and how they work. It's really helpful when you do synthetic biology to have some sense of like, okay, if I want to achieve this phenotype, what genes do I manipulate the expression of or introduce to this plant? But when you actually build these things, if you don't find what you were expecting, I think that provides opportunities to learn something about the plant that maybe you didn't know already. And in our case, if you're doing something like trying to suppress a pathway or delete a pathway, then that can be one way of finding things that are redundant and may not have otherwise revealed themselves in the wild type background. So yes, absolutely. Like genetic modifications are a great way to find redundant pathways.
That's very clear. Thanks a lot, Jen. Maybe with the two questions we have left, I want to, give it to Adrian, who asks about, the situation that happens in the base subtilis auxotroph. So the question is like, what do you suspect the reason for the escaping specific media constraint? Like, what might be the failure mode of the system that you're trying to engineer in those zeotic auxotrophy?
Yeah, we have a ton of theories about this, but no actual data to tell us what's happening. My suspicion is that the bacteria in the soil environment are entering more of a persister state. So we don't see them actually bloom. If you track CFU in the soil over time, it actually does decline. It's just that like they don't decline as rapidly. Like there's no difference in how quickly they decline with and without the plant that produces the O-methyltyrosine. But just having the plant around does seem to help. And we think that maybe in this persister state, they're just not as metabolically active. So they're not trying to grow. They're not utilizing the genes that we've selected to put the stop codon into. And so like a combination of maybe using a strain that in this soil environment actually is trying to grow, because Bacillus subtilis, at least the strain variety that we've been using, is more of a like model microbe than it is an actual like soil isolate. Like it was isolated from soil, but like a long time ago. And then selecting more carefully which genes we require the non-standard amino acid in order to produce would be my other suspicion, but I don't know. And I think the reason maybe why it there's a difference between like just soil and soil with the plant is maybe that some of that like non-standard misincorporation of standard amino acids could be occurring if the plant is being a source of standard amino acids too. We don't know. There's a lot of.
Thanks a lot for sharing, Jen. Maybe I'll ask my own question for the last question, which is like if it's if it's kind of bit long, you can sort of skip that as well. So my question is about like, can we reuse a lot of the parts from mammalian SynBio or bacterial SynBio? So you were reusing the TET-R and some of the TET-R homologs, but in some cases, like, from myself doing experiments, sometimes they can't even be transferred between organism if there are some system in the organism we're trying to engineer itself that was, interfering or having some crosstalks. So maybe can you share that a little bit? Like, is it, not that difficult, or is it very specific to the strains that you're trying to engineer when you port the parts from non-plants to plants?
Yeah, absolutely. It's a great question because I think there's been a ton of success in moving pieces from one organism to another in order to try to manipulate that second organism, right? Like CRISPR is a fabulous example, but like the TET homologs have been used across organisms, and I think it varies depending on what type of protein or gene you're trying to move and how complicated the kind of biochemistry is that it's trying to accomplish. So in the TET homolog case, like we're using them just as bacterial DNA binding proteins. All they need to do is like find and bind DNA, and DNA is relatively similar across organisms, right? There's some heterochromatin, like there's differences in how it's compacted in eukaryotics for sure, but I think some things will translate well and some things won't. Interestingly, like when we've looked at mammalian systems, especially mammalian synthetic biology, and tried to think about like, well, what things in mammalian synthetic biology could we use in plants? It's been fun to see like how many things used in mammalian synthetic biology are actually sourced from plants in the first place, and therefore like not things that we can use for orthogonal control of gene expression in plants. Like auxin, like this, small molecule dependent dimerization or degradation is dependent on a central plant hormone, and it's very, elegant in mammalian systems where it's pretty orthogonal, but in other, thinking about using it back in plants, it really wouldn't work that well. I would encourage anyone who has a system that they're really passionate about trying to get to work in plants to just consider, like, what is it that this system needs in order to function correctly? Do I expect all of those pieces to be in plants and the environment to be correct for it? And if so, like, try it and see. I think there are certainly enough cases of things working across organisms that it's a worthwhile pursuit.
That's very clear. Thanks a lot, Jen. I think, the fact that some bacterial parts are effective, especially the TET-R and TET-R homologs, are inspiring to a lot of us who maybe start from the microbe and want to move to plants, so we can, reuse or, repurpose our understanding and the materials that we have so far. So I think that's.
I feel very strong. I feel very strongly that if you get good at engineering any biological system, whether it's bacteria or it's mammalian cells or it's plants, that actually moving into a new biological system is not that hard. You have to learn some idiosyncrasies of how that system works, but having this kind of synthetic biology or engineering framework, this way of thinking about these problems, is great and pretty broadly applicable. So I always try to encourage more people to get into plants, but really, like, make those transitions if you're passionate about it. It's a good idea.
Thanks a lot, Jen. I think that helps us wrap up and encourage people who are interested in the SynBio in general to maybe consider more on the plants, and with the knowledge and information that you have. But if you were taking the course with us on the plant SynBio, that would be a good starter for you to walk through what is out there in the plant SynBio and what would be the things you want to learn along the way if you're jumping into the field, something like that. So
Okay.
with that, I'd like to formally thank Jen for sharing with us today on the talk. Is very fascinating, and the talk will be recorded and also posted online on our YouTube channel, and we will share with everyone who are interested in this talk as well. With that, I'm gonna give the floor to Kato, just in case there's any ending parts or other announcement to do before we wrap up.
Okay, thank you so much, Ice. And I would like to appreciate everybody who has spared this time to join in this webinar. I want to appreciate Jen. Thank you so much for accepting our invitation and for this great presentation you have given to our community. And I believe our community, more especially those who have been taking our course for almost 11 weeks now, they have gotten a lot of things to learn, and they will try to implement whatever they have learned and to learn more, to continue learning more. And also, I believe when we go to the practical phase, which we are taking soon, the people who will participate in that will also try to apply whatever you have shared with them somehow, somewhere. I would like to inform you that I have been seeing a question of those ones who want to still send proposals for the native African plant. Please, we closed the proposal phase, unfortunately, because we gave it enough time. And currently, we are trying to review the proposals which you sent to us and to try to select the plant. As we shared, we shall continue communicating with you via email on the proceedings, and we shall be updating you. We shall not take decisions on our own. We shall include you in the decision making. Thank you so much. Back to you, Ice, as we close this webinar.
Yeah, and maybe if there's any parting thoughts from Jen before we wrap up, maybe.
I just want to say thank you. Thank you for spending the time with me. Thank you for the opportunity to be here. It was really a pleasure, and I wish everybody the best of luck, both with your sequencing project and your careers more broadly. Hope to see many of you in plant synthetic biology in the future.
Thanks a lot, Jen. Really appreciate your your wisdom on the talk today, and also the other things that we discussed along the discussion part today. So with that, maybe we can wrap this up. Please keep in touch with us for those who are in this call. If you were enrolling to our mailing list, you see more webinars from different topics that would be involving both the plants and maybe also the SynBio and also the focus in Africa as well. With that, we can formally close this up. Thanks a lot, everyone. Goodbye.
you.